Good afternoon. If you're here, I invite you to grab a seat. We have a few more seats in the front here, and we have a copy of the flyer, the report, so I invite you to sit down for the section. So good afternoon. My name is Win, and I am the director of American Academy of Microbiology. Welcome to the Early Microbial Life Lunch panel. So thank you for joining us today, and it's my pleasure to welcome you today. We gather here today for this section for two reasons. First is to celebrate the launch of our Early Microbial Life, our past, present, and future project report that the Academy released earlier this week. This is an effort that we have put in the last 18 months, and the panel will help to share with you more detail about this. And the second reason is the celebration of the 25th anniversary of our partner organization, the Gordon and Betty Moore Foundation, who provided the support for this project. It all started about 18 months ago when John Case, project director of the Moore Foundation, and I have a conversation about the origin of life on Earth and the many unanswered questions to be explored. And I was fascinated about this topic when I think about the fundamental question and the fundamental knowledge that can answer through the studying of these origins of life and the potential application of this knowledge to inform us about how life evolved in other planets, as well as how life adapt and change here on Earth. So it was the reason why we started this project 18 months ago and bring us to the publication of the report today. Our goal is to bring together the expert in diverse field of science around the world so that we can dig deep into this topic and explore what we know and what we don't know, and how we can design effective next research topics project to answer this question. In the past year and a half, it was fortunate. I'm very fortunate to work with amazing steering committees, leading by Dr. Von Cooper and Dr. Mike Lynch, and many excellent scientists around the world on this topic. Some of them here today. And we want to share with you what we learned from this topic in this panel. It's a snapshot of what the full report that we published. And we have a few flyers that you can download the report. And this is a 200-page report, in-depth, and this is something we highly recommend. So I invite you to read the report. I hope you enjoy the section. I'd like to thank the Moore Foundation for the partnership and for many people who have involved to make this project possible. So now I will turn to Dr. John Case from the Moore Foundation to offer some remarks on behalf of the Gordon and Betty Moore Foundation.
All right, good afternoon, everyone. Thank you so much, Win. So I'm John Kay, a program director at the Gordon and Betty Moore Foundation up in Palo Alto. So pretty short trip down last night. And on behalf of the Foundation, the Academy, and ASM, I'm pleased to add my welcome for this special event today. As Win mentioned, today's discussion will reflect on the culmination of several fantastic workshops that I had a chance to observe that dove into questions of deep evolutionary time, seeking to assess the state of play with several of the major transitions of microbial evolution and to brainstorm possibilities for how to pursue further research in these areas. As you'll hear, the report brings the biological lens to the forefront and demonstrates how integrating expertise across microbiology, geobiology, and evolutionary biology holds potential to unlock discoveries that no single discipline can achieve alone. Today also represents an opportunity to celebrate the Moore Foundation's 25th anniversary, along with 4 other events this year that collectively reflect the diversity of our Foundation's programmatic areas, from science to environmental conservation, and to a focus on preserving what makes the San Francisco Bay Area unique. So here, given our Foundation's interest in scientific discovery, The Academy's report and the special session today serve as an opportunity to better understand new research avenues and opportunities. In addition, we're glad to be able to tie this event to our anniversary because as part of our core approach, we value the opportunity to support experts to envision what the frontiers of science could look like over the next 25 years. It also provides an opportunity to celebrate each of you and your positive contributions that you're making to science writ large. So in that spirit, we're pleased to provide some thank you mementos to the panelists, you'll get them later, and to the great staff at ASM and the Academy that helped make this event and of course the entire Microbe Conference come to life. So let's hop from mementos to big dollars for a moment. So why has the Moore Foundation been interested in the history of microbial evolution? So the Foundation's interest in early microbial life is not coincidental, but in many ways is an outgrowth of years of support of basic research on microbial ecology, physiology, and evolution as part of what I view as an ongoing revolution in the microbial sciences. I presume all of you will agree with that. And we will, through our now concluded Marine Microbiology Initiative, our ongoing Symbiosis and Aquatic Symbiosis Initiative that focuses on microbial symbioses and other grant making, the Foundation has supported upwards of $500 million of microbial science research. And we will continue to scan the horizon for new and equally exciting opportunities. So to conclude, a brief word on Gordon and Betty, our founders. So I've been at the Foundation for 18 years, so somehow that happened. And so I've seen how the organization has grown and found its footing along the way. And though some of the components have changed over my time and over the past 25 years, the most fundamental elements have not. So this is truly a remarkable achievement of the durable vision that Gordon and Betty laid out by, when they set up the philanthropy in their name. So we will continue to be guided by their simple but powerful principles, and I just want to mention two of them today. So the first is the imperative of finding important leading-edge scientific niches where the scale of the Foundation's contributions could make a substantial difference. The second is to create synergies whereby the whole is greater than the sum of the parts. And I hope you'll be able to see those principles reflected in today's discussion. I'll end with deep gratitude to Wynn for leading this effort on behalf of the Academy. My thanks again to the ASM staff. Some are here, some probably not here at the ASM. And my sincere appreciation for the heroic efforts of Vaughn and Mike as co-chairs of the project, and for the entire steering committee, and also for all the dozens of scientists who participated in the workshops over the past year and a half. It really has been a remarkable set of conversations. Looking forward to the synthesis now. So thank you so much, and I'll turn it to you, Vaughn. Thanks.
Good afternoon. Really delighted to see so many of you here. I stand between you and what you really came to see, which is going to amount to basically a highlight reel of 4 of the leading participants in our workshops. Although really, it was sort of, 3 dozen all equal experts spanning the fields. I really want to echo my thanks to John and the Moore Foundation. Those principles were completely transparent throughout this entire process. They sort of realized that this area of origins of life and early microbial life was an area where a strategic investment could bring together people from across the sort of horizon of the sciences, work together and identify areas for potential breakthrough. So just really quickly, the goals were to identify the scientific gaps, to explore opportunities, for future scientific research and implications, and I think most significantly, now wearing my ASM hat, I guess I'm a company man now as incoming president-elect. But no, in all seriousness, we, ASM, really wish to provide a home for this community. So, Mike and I were chairs of the steering committee, along with Susanna Coelho from Max Planck, Betul Kachar from Wisconsin-Madison, Will Ratcliffe from Georgia Tech, and Paul Turner from Yale. And there were 3 colloquia, 3 of the most kind of electric meetings I've ever had a chance to be a part of. The first colloquium was really on the scale from understanding the core questions about the origins of life all the way up to the last universal common ancestor of life, Luca. The second was a colloquium on from the last universal common ancestor to the last eukaryotic common ancestor. So as Will Ratcliffe said, from Luca to Lica. And we really focused on all of the potential branches along the way. And then the third was about the origins of multicellularity. Our goal, again, was to activate and really broaden the evolutionary microbiology community at ASM around these topics. So I really want to say that this highlight reel is going to be great, but there's so much more. One of you said that this report might even be a great undergraduate textbook. It's a terrific bit of work, and I want to say it was really only made possible by the heroic work by the Academy staff, Wen and Donna Lynch-Uner, who I don't believe is here. So with that, I want to call up the first speaker, Tom Williams from the University of Bristol in the UK. So we want to, the next. While we're doing this, I want to say, because this is intended to be a panel, please hold your questions to the end. Please write them down. You're going to have them, and stick around to the end. And also, this is being recorded, so please use the microphone when we get to those questions.
So hi, everyone. It's wonderful to be here. Thanks, Fawn.
Sorry, Beth, my bad.
That's okay. I just moved that, so you just moved. That's cool. So yeah, it's wonderful to be here. I'd like to thank the organizers really for the opportunity to take part in this wonderful initiative and be involved in doing the report, as well as talking to you about some of it today. So yeah, I'm really excited to be here to talk a little bit about the origin of life and kind of the early evolution of prokaryotes. And I'm not going to be able to give you like a very full treatment of those topics in the next 10 minutes, in part because I'm going to shoehorn a little bit of my own work into the talk as well. But what I will try and do is give you some take-home messages really from the colloquium and some of the discussion points that we had. And I think the kind of starting point here for talking about this is that really people have been studying the origin of life from at least 2 different approaches, right? So there's the bottom-up approach. That's where chiefly chemists try to identify plausible pre-biological ways that, you know, life's building blocks could have been assembled on the early Earth, or even how, you know, primitive replicators could have started replicating. And then the other approach is the top-down approach. That's where, you know, people like me, so evolutionary biologists, we're trying to compare lots of different organisms through our life today, and using their core features, try to ask what those features mean about what early life could have been like. And while these are 2 different communities, I think as you'll see, it's really interfacing between the 2 where you actually get progress on the origin of life. So from my perspective, you know, the starting point for trying to understand early evolution is the tree of life reconstructed from modern genomes. This is the sort of increasingly iconic picture from Hug et al. And what you can see here immediately is why an organization like the ASM would be so interested in the origin of life, right? Because most of life's genetic and biochemical diversity is microbial. We're down here, little tip here, and then all of these different groups of bacteria here and archaea are, of course, all single-celled. And, you know, most eukaryotes are single-celled too. So how do we use this tree then to study the origin of life? Well, I guess the way that comes in is that this last universal common ancestor that Vaughan already mentioned is often considered a sort of important milestone en route from life's origins to the diversity of life that we have today. And so, you know, we and others have been interested in trying to reconstruct the evolution of genomes and gene families and try to work back from modern diversity to understand what sort of organism the last universal common ancestor might have been. And many groups have been trying to do this, but the bottom line is that we think that LUCA was actually already a rather complex organism. It would have been a prokaryote, in some ways not so different perhaps from the bacteria and archaea today. A recent analysis that we did suggested a genome of maybe 2.5 to 3 megabases, on the order of 2,500 genes or so. And so it seems that LUCA was already, actually a rod-shaped prokaryote, we think. It had an ATP synthase complex on its cell membrane, which it was using to harness energy using a proton gradient, just like in modern cells. And it was likely an autotroph, perhaps an acetogen, using the Wood-Lungdahl pathway. And also, we think that it may already have had an early immune system in the form of Cas proteins, at least, which I guess confirms or, well, suggests that early life was already interacting with viruses. And one interesting line of discussion at the meeting was the role that viruses might actually have played in shaping cellular evolution, even at the earliest stages of life's development. So a complex LUCA kind of raises this question of how life went from something presumably quite simple to the common ancestor of all life that we see today. And I think the important thing to realize here is that actually LUCA and the origin of life are two very different things. And hopefully this diagram shows you why that's the case. So in black here are the branches of the reconstructed tree of life. So that's what we saw at the beginning from the Hug et al. Study. And if it's true that many lineages have gone extinct through time, and if it's also true that we haven't sampled all of the microbial diversity that exists today, then actually that reconstructed tree of life is just a subtree within a much broader history of life. And so there's this period from LUCA here back to the root of the tree, which is where all of the sort of common features of biology must have evolved. And if you tried to do molecular clock analyses to work out how long that period was, then it seems that it wasn't terribly long in absolute terms, perhaps from 300 to 700 million years, but that's when life originated and where all of the biochemistry that we know in modern microbes first evolved. And while I think evolutionary biology actually does have a lot to say about this period, a lot of the insights that we've gained into that have come from these bottom-up sort of chemistry approaches. So I want to briefly summarize what those have told us about early life. And I think conceptually, life forms need some kind of genetic material, right, as an information store. They need a metabolism, something that they can use to grow and reproduce. And they also need compartmentalization of some kind. It doesn't necessarily have to be a lipid vesicle. And that's because you need first to concentrate important biomolecules for use in metabolism, but also because you need to create a kind of cohesive evolutionary unit, something which can evolve. And so these are probably some of the key features of early life. And we can ask, what were the earliest, if you like, physical forms that each of these features of life took? And so when it comes to the genome, the RNA world hypothesis is probably the leading explanation for what came before DNA. So RNA, of course, is very similar to DNA, and it's just as good an information store. The sequence of bases in RNA can store genes, just like in DNA. But RNA is also different to DNA in that it's more flexible. It can fold up into different shapes and catalyze different reactions, like proteins do in modern cells. So the hypothesis, I think, for the first genome is something like this. There was a pool of perhaps RNA molecules of random sequence and structure, and through processes of evolution, including selection for self-replication, that pool was perhaps enriched in molecules which could replicate themselves, and that might have been the first genome. What about the beginnings of metabolism? Well, I think the most striking result from this field is actually that some of the core reactions that we think took place in LUCA, like glycolysis, perhaps like the Wood-Lungdahl pathway, some of their steps can be catalyzed on transition metals. So it's possible that perhaps on a mineral made of iron or something similar, some of these early reactions could have taken place. So the hypothesis there is that the first life forms harnessed processes that were already occurring naturally or pre-biologically and perhaps sped them up. And then finally, what about compartmentalization? So there's a number of different ideas about this, and for reasons of time, I'm just summarizing one of the interesting ones here. And this is the idea that the first compartments could have been perhaps in the walls of alkaline hydrothermal vents. And the best studied of these is the Lost City vent in the middle of the Atlantic today. That's a photograph of that. And what's interesting about this scenario is that because you would have alkaline fluids coming up from within the vent, and on the early Earth, probably quite acidic water, you would have had a proton gradient at that interface across the wall of the vent. And it could have been there either in lipid vesicles or even in inorganic compartments, according to some authorities, that this could have been an early proton gradient that could have been harnessed by early metabolisms. In much the same way, perhaps, as we think the ATP synthase molecule was doing in LUCA and as it does today. So I'm going to finish there. I'm just going to say that, biology can do a lot more, I think, than it has done so far in terms of working back to those earliest stages of evolution. And I think we're going to hear more later in the session about some of the ways that evolutionary theory can perhaps constrain our ideas for the origin of life. So thank you very much.
Thanks very much, Tom. Amazing. So I'd love to call up Dr. Puri Lopez Garcia from CNRS in University Paris Sud. Oh. Yeah, here. You want to help? Yeah. Thanks.
So good afternoon, and I want to join Tom in thanking the fantastic group of people who have helped this symposia to be organized and lead these interesting discussions. So I'm going to talk about the origin of the eukaryotic cell, eukaryogenesis, that represented a unique major evolutionary transition that led to the evolution of complex cells having an endomembrane system and a nucleus, flagella, complex cytoskeleton, and mitochondria from simpler ancestors, and that led to a radiation, a wide diversification of organisms and lineages, most of them corresponding to unicellular eukaryotes, protists, but some of which led later to several independent origins of multicellular organisms. We know that during this process, endosymbiosis was very important, as illustrated by the origin of plastids and eukaryotic photosynthesis and mitochondria. We now know that plastids are closely related to a newly detected order of cyanobacteria, the Gliomargaritales. By contrast, with an extended taxon sampling and more sophisticated models of sequence evolution, we have not been able to identify a closely related group of alpha protobacteria to current mitochondria. We are still looking for the closest prokaryotic lineage to mitochondria and associated to the alpha protobacteria. What we now know as well is that Asgard archaea took an important role in this process, eukaryogenesis. This is a group of archaea that have been known for some time based on amplicon sequences, but they became famous 10 years ago when the first metagenome assembled genomes were assembled, and they revealed the presence of homologs of eukaryotic genes involving a wide variety of processes, including cytoskeleton and the membrane remodeling, more genes in common with eukaryotes than other archaea. And phylogenomic analysis started placing eukaryotes within this group of archaea, notably within the Heimdall archaea. We also know that these archaea are strict anaerobes. They live mainly in marine and freshwater sediments, microbial mats, some are thermophilic and live in hot springs. They are inferred to be mostly heterotrophic and hydrogen scavengers or donors, which in the kind of environments where they live, it implies that they are most likely involved in syntrophic interactions with other members based on hydrogen interspecies transfer. And this is actually somehow validated or confirmed by the first cultivation of two members of this group that are obligate syntrophs, either with methanogenic archaea, with sulfate-reducing delta protobacteria, or the sulfobacterota, or both. And there are now other members in culture also obligate syntrophs. So with all of that, we have been shifting our views on eukaryogenesis over time from simple linear models at the beginning when prokaryote and eukaryotes, this dichotomy was discovered, to models where eukaryotes were a third lineage in the tree of life, a third primary domain, to models now that suggest that eukaryotes are symbiotic mergers of bacteria and archaea. Such symbiotic models already existed in the late 20th century, but they have really become mainstream after the discovery of Asgararchaea. And symbiogenetic models of eukaryogenesis can easily explain the chimeric nature of eukaryotes that have archaea-like informational processes, genes and proteins involved in DNA replication, transcription, translation, but bacterial-like bioenergetics related to the mitochondria and membrane phospholipids. So when did eukaryogenesis occur? Both the fossil record and also molecular dating converge to suggest that this process, which may have been long, started slightly after or perhaps coinciding with the great oxidation event, the oxygenation of the atmosphere, and where in which kind of environments, most likely in redox transition environments such as microbial mats or shallow sediments. The oceans at the time were anoxic. And in this kind of environments, Asgararchaea could have met the facultative aerobe ancestor of mitochondria. How did eukaryogenesis occur? And this is where most models diverge. Symbiotic models can be roughly classified in three major groups. Most of them propose only two partners, one Asgararchaeon as a host for the ancestor of mitochondria that was somehow engulfed. There are models that propose transient symbiosis with other bacteria, either sulfate-reducing delta protobacteria or with different bacteria prior to the final acquisition of the mitochondria. And there is at least one model, the syntrophy hypothesis, proposing three stable partners, one Asgararchaeon that ferments and becomes an endosymbiont within a sulfate-reducing delta protobacterium, and then the ancestor of mitochondria, a facultative aerobe, and also a sulfur oxidizer in this context. There are many open questions regarding eukaryogenesis. One of them is how did the membranes evolve? And in particular, models that propose an Asgararchaeal host need to evoke a transition from archaeal to bacterial phospholipids that is not easy. So did this process, this phospholipid transition occur, or there was somehow a too important fitness cost, and this did not happen? And then could a bacterial host with the continuity of bacterial membranes have been something important? And this would perhaps fit well with the observation that eukaryotic viruses are bacterial-like and not archaeal-like, as would be expected if the host was an archaeon. How did the nucleus evolve? This is an open question that many models don't address, which were the selective forces and the mechanisms involved. It's very open. Was phagocytosis needed for the incorporation of the ancestor of mitochondria, or some other kind of process, such as the protrusion that can be seen in Asgararchaea because they have a complex actin cytoskeleton in particular? But then similar processes exist in bacteria that can have very different shapes and also a developed cytoskeleton. And unlike archaea, which don't have complex endomembranes, many bacteria do have endomembranes, and even some of them do have true phagocytosis. Did mitochondria arrive early or late? That's another problem. Some groups, scientists have been studying this by looking at the average stem length of genes of prokaryotic ancestry in the ancestor of eukaryotes and the distance with their closest outgroups. And this analysis suggests that alpha protobacteria came relatively late, but this analysis also bring up to the table an older and more diverse contribution of bacterial genes to eukaryotic genomes. How did these genes arrive to the LICA genome? Did they arrive by horizontal gene transfer from bacteria in the same environment, or were there somehow transfer from bacterial endosymbionts, either permanent or transient? This is another open question. We can try to make progress in all these questions by applying phylogenomics to better define the prokaryotic ancestry of LICA genes, studying the molecular and cell biology in particular of prokaryotic lineages that seem related to eukaryotes, and better understand the microbial ecology of prokaryote-prokaryote symbiosis. But what is fundamental is to have now an integrative and critical view, and to propose detailed models on the origin of eukaryotes that propose plausible processes that can account for all the observations, the observed patterns, in a realistic ecological context. Thank you very much.
So next is Julia Schwartzman from just up the road from the University of Southern California. Just want to comment, you're probably all familiar with the challenge of building phylogenetic trees in various parts of your research. And what I learned from these symposia is that I had no idea how difficult this kind of work is, and yet how valuable it is.
Fantastic. Well, thank you, Vaughn, and thanks, Nguyen, and all of the organizers. It was really an exciting time to come together and talk as a group about the evolution and mechanisms governing multicellularity. I wish I was at the other two meetings as well. They sound phenomenal as well. So my job is to represent this large group effort that happened back in January and try to represent our field. Okay. So this is great because it's already been introduced by Tom and Puri, but we have the Tree of Life, and one of the really exciting things that's come up recently is that truly multicellularity not only arose multiple times in the eukaryotes, but also in bacteria, and recently people have discovered multicellular archaea. And so we're thinking all domains of life. It's a little hard to see on this tree, but multiple independent origins really pose a fundamental question, which is taking a step back, how do we think about this as one framework, as opposed to staying kind of siloed in different fields grouped around individual domains? Okay. I just want to convince you, I just want to convince you that indeed there are commonalities across the domains. So here's an example of archaeal multicellularity. I'm going to show you an example of bacterial multicellularity, if the video will play, and eukaryotic multicellularity. And all of these are early multicellular forms, or maybe not what we would call complex multicellular life, but emergent new multicellularity. Okay. So a very useful framework to think about the evolution of multicellularity, especially in a really general context, is evolutionary transitions. It's already been brought up so far. And really, I want to focus on this transition between single cells to multicellular groups, and from multicellular groups to individuals. A theme that came up during our group discussion, and that's in the report, is really multicellularity is an adjective that modifies a noun. So we're thinking you don't have to argue about what is and what is not multicellular. You can have aspects of multicellularity that can kind of emerge and can manifest as different steps in this transition. And that way of thinking, I think, gets rid of a lot of semantic arguments that one can have in the field. Okay. So thinking about multicellular groups, groups are really different than individual cells, because a group can come together and have emergent properties and cooperate and do something greater than the sum of parts. Examples of groups, it's not entirely, doesn't necessarily have to be just one particular species. You can think of a group as a consortia, for instance, these sulfur cycling bacterial granules. You can also think of consortia spanning domains, so archaea and bacteria can together do something collective that would be considered a group in this framework. Biofilms also fit in the context, so this is really a big tent, all-inclusive. We can think of all of these as having multicellular traits and properties. Maybe not all the same traits and properties, but at least aspects. And of course, we can think about this encompasses multicellularity that emerges within the eukaryotic domain. I want to emphasize that for multicellular groups, these are often transient forms of multicellularity. They're ephemeral. Biofilms form and disperse. The choanoflagellate rosettes come together only under certain environmental contexts, and so this is a really important aspect of this transition. The properties of coming together, a real important property of multicellular groups is phenotypic variation, or if it's a consortia, the variation between different genotypes that bring different functions to the group and can create an emergent function. We can think of phenotypic variation in biofilms as potentiating long-range signaling in the group. You can also think about structural divisions of labor, or in a mixed species biofilm, such as the granules in bioreactors, you can think of really complex metabolism that's suddenly able to be achieved by coming together and forming a multicellular group. In contrast, if we're going to think one step up the transition in individuality, multicellular individuals have specialized cell functions. So these are things like Volvox carterii, or Streptomyces silicolor, Myxobacterium, Anabina. All of these forms of microbial life that are multicellular, the cells are doing something distinct that's helping the individual reproduce as a group. One of the really interesting themes that emerged that we talked about in our colloquium was the fact that I would say maybe even 10 years ago, it was thought that to be a multicellular individual, maybe what had to happen was that there were new genes that had particular functions. But what's really emerged is that there's very little that's been innovated, very little that's new that is in multicellular life when you're starting to compare to extant unicellular relatives. So for instance, the comparison between choanoflagellates and other metazoans revealed that really the differences are regulatory more than structural. So eukaryotic cells can come together and rewire their regulation and achieve something that's greater and more complex. And I find this really, really fascinating because that suggests, if we're going to think cross domains, that maybe the way forward is to think more as modules and more as problems solving different regulatory challenges. So shifting gears slightly, I really wanted to emphasize a theme that came up during our colloquium, which was, and it's actually, I think Puri also kind of echoed this, ecological context. This is close to my heart. I'm in part, I wear a microbial ecologist hat. And so when we think about the evolution of multicellularity, at least if we're going to think of selection as the evolutionary force, this really only makes sense in light of the ecological context in which evolution took place. And so this is a really beautiful review that was written by Will Radcliffe and his group, kind of outlining the different drivers, ecological drivers through which multicellularity might be selected for. These can be things like the avoidance of predators, the ability to resist stress, like would happen in some biofilms, potentially cross-feed, like we talked about with consortia, compete, disperse, make propagules. And so all of these can be binned in kind of conceptually distinct groups. It's really great to have a framework like this because it becomes experimentally tractable in the lab to take model organisms or even organisms from the wild and create a really defined, controlled environment where we can understand how each different driver can select for multicellularity and what the trajectory might be for different microbial forms across the tree of life. For instance, recent work evolving, I think it was E. coli, to grow as multicellular clumps, or even older work by Vaughn Cooper here on the evolution of Pseudomonas and Burkholderia cepacia biofilms are great examples. You can do this with you can do this with microbial life across the tree. So cyanobacteria, people have thought about as experimental models for experimental evolution. Of course, Will Radcliffe's work on yeast and cyanobacteria. So getting closer to my frame of expertise. So one of the things that, and maybe this is my bias, I find really fascinating, you can think about ecological drivers on their own and without the larger context of nature. But then one thing that I've been really excited to see people go out and do is looking in nature and looking at combinations of drivers and the patterns that you see in nature. So diel rhythms, differences in, I'm showing you a fantastic example from Thibaut Brunet's group on, I guess it would be the left, looking at the evolution of multicellularity in choanoflagellates in these splash pools where there's daily evaporation. You can think about biogeographic patterns where you're looking at the emergence of multicellular groups across the ocean with different environmental gradients. And then my group is thinking about the microscale interactions that can drive the emergence of multicellularity in heterotrophic bacteria that live in coastal marine environments. So just to take one step back, all of this is really, really interesting, but of course, if we're going to think about the deep evolutionary origins of multicellularity, we can think about this experimentally and we can come up with scenarios that might be relevant, but what would be really great would be to look back into the fossil record. And one really exciting thing that we heard about in the colloquium, and it's in the report, is this idea of microbial fossils. I hadn't been following this field as closely until I went to the colloquium. I really encourage you to don't sleep on it, look at the literature, it's amazing. There are a lot of microbial fossils out there and examples of potentially early multicellular life. And what's really striking is often it's just one or two examples that have been characterized, but there are fossil beds across the world where you can go and really we're very early in the sampler's curve of being able to characterize these forms of multicellularity. And if we could, so most multicellular life that we know about, I think, is represented in the Phanerozoic and Proterozoic, but we might even be able to look farther back into rock formations that are representative of the Archean and find really early examples of multicellularity, and that would be truly exciting, because then we could tie the kind of ecological and lab experimental work into something that's more grounded in the geologic record. So I wanted to conclude with the thought that, at least for our particular field, and I think in several of the others, this is a truly interdisciplinary field. I think it's hard to bring all of the people who are at our colloquium into one room at a particular conference, so it was a really special opportunity. And you know, to move forward and to think about the mechanisms and evolution of multicellularity, I think is a truly interdisciplinary problem that requires paleontology, theory and computation, experiments in the lab, comparative biology, synthetic systems, and of course, ecological studies. And I hope you read the report and our ideas about going forward as a field. Thank you very much.
Thanks very much, Julia. And so it's my great pleasure to invite Mike Lynch to come up for the last section. And just I want to highlight, none of this really would have been possible without Mike's vision within this field to help identify these individuals and bring them together, and also to motivate us all to think about the one true unifying concept that we really need to dig into, and that's evolutionary theory.
Great, thanks a lot, Yvonne, and my predecessors up here, and all the other folks that contributed to this report that's come out. It was a really amazing experience, I think, that we all had over the past year or so. What I'm seeing out of this is the emergence of this new field, what we call evolutionary cell biology. It's we're not allowed to use the word integration anymore. I think that's one of the forbidden words now in the US, but we'll call it interdisciplinary thinking. And we're trying to bring together all these different fields to deal with these really difficult problems that can't be handled by any one subdiscipline. When I think about evolution, I think there's three dimensions of that dictate how evolution can and can't occur. So most evolutionary biologists are thinking about the things at the bottom of this diagram here, things going on outside of cells, finding, you know, resources, avoiding bad things like predators and pathogens, finding a mate of the right type and the right species, and so on. But everything that we're built out of starts at the cellular level, and of course cells are built out of molecules. So there's sort of a natural history that goes on inside of cells, and there's certain rules that we have to obey there. We're not allowed to break the laws of biophysics. There's a lot of weird things in biology. I'll mention a couple of them later, but there are historical contingencies that were laid down over 4 billion years ago, and there's no way going back now. And then molecular stochasticity turns out to be a really important source of variation, that it actually operates against selection because it eliminates the signal from the genetic level. But ultimately what can happen in evolution, what natural selection can and can't do, is dictated by what I call the population genetic environment, which has these 3 dimensions. Mutation, of course, is the source of all variation upon which natural selection operates. Recombination reassorts variation within and among chromosomes, and random genetic drift is sort of the noise in the evolutionary process that dictates how efficient selection can be. These are sort of the 6 commandments of evolutionary biology. Most of you know these, but the point here is that these deep problems, such as the origin of life, the origin of eukaryotes, they're all really problems at the genetic level and really at the population genetics level. And these are things that people in population genetics have sort of been hands-off on, dealing mostly with sort of neo-evolutionary problems rather than these deep problems. But they're ultimately, these are the mechanisms of evolution. So you know what mutation does. Random genetic drift is maybe somewhat mystical to all of you, that this is a ubiquitous force in evolution. It's caused by finite numbers of individuals and populations, but more importantly, because all our chromosomes, all our genes are glued together on chromosomes, and that causes selective interference, that causes noise. So the efficiency of natural selection is dictated by the power of random genetic drift, which you'll see in a second varies dramatically among lineages. Important thing to realize that we forget about all the time here is that a permanent response to natural selection absolutely requires genetic variation and requires genetic change. And evolution is not a Shazam process. Evolution occurs by incremental changes. Single mutations come in and navigate a very difficult sea of random genetic drift and different genetic backgrounds and so on. If we want to understand evolution, we need to start thinking that way. All of you know that evolution is a process of descent with modification. We work with what we were given from our predecessors. And then finally, the second big engine in evolution is not just the divergence of individual lineage, but the creation of new lineages that are reproductively isolated from each other, and that's based on genomic incompatibilities. Now, we're interested in this issue of the population genetic environment because all the dimensions of it vary by about 10,000-fold across the tree of life. So on the left here, we're looking at what we call the effective population size of organisms. That scales on a log scale of a minus 0.2 power. So if you increase the size of an organism by 5 orders of magnitude, you also increase the power of random genetic drift by 1 order of magnitude. And the middle panel is a summary of a bit old summary, but the pattern is held up. There's an inverse relationship between the mutation rate at the nucleotide level per generation and the effective population size. Things that have low effective population sizes, natural selection is not as efficient at driving up replication fidelity. And then to the right is how the recombination rate scales with genome size. It's a nice inverse relationship there as well. And that's simply because in eukaryotic organisms, as you increase the size of a genome, you're not generally increasing the number of chromosomes, you're increasing chromosome number. There's typically 1 crossover per chromosome arm, and so that will give you this nice relationship here. Now, to give you an idea of why this becomes important, here's this plot again of the relationship between effective population size and the size of an organism. And bacteria up there in the upper left, and then we get into unicellular eukaryotes, and then we move down into multicellular species. And why this is important is to just take a look down at the bottom there, which is where you live. Mammals have generally very, very small effective population sizes. So the human effective size until recently is about 10 to the 4th individuals. And the way to think about that is the power of genetic drift is the inverse of this number, 10 to the minus 4. What that means is if a deleterious mutation comes along with an effect less than, a harmful effect less than 10 to the minus 4, natural selection can't weed those out. And likewise, if it's a beneficial mutation with an advantage less than 10 to the 4th, or 10 to the minus 4, we can't take advantage of that. And then we move up here at the top, we're looking at bacteria. I've got 10 to the 9th individuals there. That's a genetic effective population size. This is not the absolute number of individuals in a species. This would be a tiny pile of bacteria, but that's as good as it gets for organisms on this planet. But what this means is that bacteria can take advantage of mutations with effects as small as 10 to the minus 9. This means that there's evolution is much more granular when you move down this panel here from microbes to vertebrates. So all the same rules apply, but the magnitude of the different effects at the genetic level change dramatically. This leads to this thing that we call the drift barrier hypothesis. So imagine you have a bunch of organisms all under exactly the same selection. The target up there at the top is the biophysics limit. Maybe this is the maximum catalytic rate of an enzyme or something, or the minimum error rate if you're replicating DNA. And as you move to the right, you're going to, so selection is uniformly pushing things up, but as you move to the right, population sizes are getting larger and larger, natural selection's more efficient. And the mean phenotype changes, even though nothing has changed in terms of the strength of selection. So if you were to look at species under identical forms of selection, but simply had different population sizes, you would see a gradient in the mean phenotype across the tree of life that you're looking at. So this is an important result that comes from a lot of math and so on, and that you're not going to want to hear about today in population genetics. But the point is that this underlies all mechanisms of evolution. Now, origin of life, you've heard we've made a lot of progress on this, but this is a peculiar field, the origin of life field. There's national, international meetings every year, and there's almost no evolutionary biologists at these meetings. It's really dominated by chemists and geologists. Beautiful work. Folks are concentrated on trying to figure out plausible places where life might have gone off the ground. We heard about one of these alkaline hydrothermal vents, but soda lakes is another possibility, and there's a number of different geological possibilities. And this has taught us a lot about how life might have evolved. But ultimately, getting life off the ground is a population genetic process. Maybe there was an early RNA world. There was certainly an early world in which things are much simpler than they were today, and you had molecules like this. And one of the biggest challenges of getting life off the ground is trying to figure out how to grow a genome. And non-enzymatic replication of RNA, for example, has an error rate of 0.1 per base. That's not good, right? So if you have an offspring with only 10 bases, it's got a mutation in there. So the challenge here in population, from a population genetics point of view, is how could we grow genomes and co-evolve functional aspects of the genome with parts of the genome that dictate replication fidelity. Another problem we heard about a little bit earlier as well is genomic parasites. Probably parasites were around from the very, very dawn of life, even before cellular life. Any molecule can take advantage of public goods at any point in time without producing them is going to have a selective advantage. So this is a thing that evolutionary biologists studied a lot, but not really in the context of the origin of life. You all know about the genetic code, and people talk a lot about the genetic code and which amino acids came in at which time and so on. But nobody's really thought about this in terms of evolutionary mechanisms. So we might have started out with a completely anonymous code. There would have been triplets and so on, but maybe initially any old amino acid could have been loaded in. But then somebody, one individual in some population decided I'm going to use GAA for glue in the future. That then has to pass through a whole population over time to really cement this as a permanent change in one of our ancestors. The origin of the ribosome, this is one of the big mysteries of the origin of the cell, is the ribosome. Really complex thing, 100 or so proteins and sort of an onion skin model. This might be one of the epitomes of over-design and underperformance of a molecule, but we're stuck with it. And it's entirely possible the ribosome initially started out as a parasite and then eventually became integrated into the, there I go using that word, I don't know what, let's just leave it at that, and became part of us as clearly as an obligate mutualist early on. Now, moving on to the next big problem that we tried to tackle in these series of colloquia is the origin of cellular complexity. And there's this myth among biologists that there's a never-ending chase to make things more and more complex. And you're all working on bacteria, or many of you are, and you know that most of the world did not go down that route. And maybe there's a good reason for that. But when we got to eukaryotes, things did in fact become very, very complex. That's a model sort of the genome up there of eukaryotes versus prokaryotes, the addition of introns, mobile elements, and so on. Most of these things are not intrinsically advantageous at all, but they were able to creep into eukaryotic genomes based on the reduced power of efficiency of selection. Enzymes, most enzymes are operating not as monomers, but as multimers. And there's many enzymes we now know of that in some species are dimers, others are tetramers, others are trimers, and so on, having exactly the same function. So a big question here is why this increase in complexity of molecular architecture, in particular in eukaryotes, the different subcomponents will often be encoded by different genetic loci. These would be called heteromers as opposed to homomers. And then even if we're thinking about something that is complex, but in some sense seems kind of simple, is the origin of the plastid, the origin of mitochondrion. This must have started out as a single event in a single individual, possibly initially as a parasite again. And the question is, how can this kind of process go on over evolutionary time and get permanently integrated into the ancestor of all? Here's one other example, specific example of origin of molecular complexity that seems to have really gotten us nowhere. So this is a strange molecule called RNase P. Its main goal in life is to cleave a single phosphodiester bond and transfer RNAs. And in bacteria, it's got a RNA part and a protein part, but just two parts. And when you get into eukaryotes, you get this big set of new embellishments, 5 to 10 new proteins. Same function as in bacteria, but a much more complex molecule. There's no evidence that the enzyme has become more efficient, has enhanced speed, or is more accurate, and so on. But these kinds of things happened repeatedly when we moved into the eukaryotic domain. I think this is the last example I'll just show. This is one of the strangest things in all of biology, this thing called ATP synthase. It's sort of like a turbine on a river, except instead of water, we're using protons to go through ATP synthase, and they twirl the what's called the F1 component up there, and one complete revolution. Rotation causes the production of 3 ATPs. And what's become interesting over the past few years, there's a ring in the membrane there. This is embedded in the membrane. It's called the C ring there. There's tremendous variation in the number of components in the C ring. And why this is interesting is from a bioenergetic perspective, because you have to load into each one of these a proton to get a complete rotation of the FO to get 3 ATPs. So up on the top, we have a C8, so it only takes 8 hydrogen ions to produce 3 ATPs. So you can think about that as sort of the Prius of the ATP synthase world. And then down at the bottom, we have things as large as 17 subunits. So how do these complexes grow, and why on earth would you even want to do this? Because you've got a much less efficient motor going on. This is a really strange molecule. This is like building a hydroelectric plant on a river and then just discovering later on there's no water in the river. So now we have to pump above the dam to get our electricity out. So that's what's going on. And finally, I won't say too much about multicellularity. But I will emphasize this isn't just an issue of eukaryotes. These are two examples of multicellularity in bacteria, Lampropedia up there and Merismopedia. One's a beta-proteobacterium and one's a cyanobacterium, and they They have very similar sort of postage stamp-like colonial structures. And one of the things that Julia just mentioned is one of the things we've learned about evolution and multicellularity is that most of it's not due to the origin of new genes. In fact, it's often due to what we call the subfunctionalization of pre-existing genes, so that one copy and another copy may be expressed in different tissues. This is a problem. This has happened, but it's a problem for biology, because if one of your copies decides to misbehave, you've got a problem like cancer or something. It gets turned down in the wrong tissue and so on. There's a number of other issues here that are relevant to the bacteria, of course, have sex, but we eukaryotes have meiotic sex, and this has led to many other things, including sexual selection and so on. But all these have been addressed by population geneticists, but mostly from the standpoint of vertebrates and land plants. So there's a lot of new areas to really expand the whole field of evolutionary theory into, from the standpoint of the questions that we've been asking. I'll just finish up by mentioning this, sort of the big picture, the big financial picture. So physicists and astrophysicists are really well organized. They get together in large groups of individuals and convince people to give them lots of money. So just to give you an idea where we are, you've all seen these beautiful photographs from the Webb telescope. That cost about $10 billion to make, and it's costing about $200 million a year to operate. The focus here is on the origin of the universe, right? And, you know, it's an interesting question. There are groups of physicists think there's a multiverse out there, so there isn't one universe. So, you know, where do we stop? But we'll just leave it at that. There's a movement now to send humans to Mars. How much would that cost? About $500 billion. The little robot that we've had up there called Rover is about $1 billion project. I think this is now mothballed, but there was a motion to produce a new super collider superconductor to look at particle physics sorts of problems, $11 billion, and if it ever gets resurrected, it'll be more than that. Okay, how does this all compare to how much we put into evolutionary biology? The budget of the Division of Environmental Biology at NSF, which is where most projects related to evolution are funded, the annual budget's $100 million. It's slated to be lopped off by about 50%. That'll be down to $50 million. And of course, we don't even know for certainty if NSF will exist a few months from now. But look at the scale here. And so the point here is that, you know, we're out there exploring space, trying to find the origin of the universe, and we have these basic mysteries about the origin of us, the origin of life, and we could make a lot more progress for, you know, a lot smaller budget, and it would have a lot of impact on many areas of biology you've already heard of. Synthetic biology would be another one of these. So what we're trying to advocate here as a group is sort of a moonshot for biology, is try to organize ourselves, think about what we could do if we organize ourselves like physicists and astrobiologists and so on do. And that's it for today. Again, I'd like to really thank Vaughn and my other colleagues up here that this book really, that we were able to put together in a really short period of time, I hope will be really useful for people. Thank you.
Okay, so thank you for patiently holding questions. We are close to time, but we are glad to take them. If you have a question, please come up to the microphone, and Mike, you can use that seat and microphone if you want. Fire away with the question, and if you know who you're directing it to, go ahead and name them, but otherwise, we'll just see how we handle it. It's on.
It is on. Oh, okay. Thank you. Thanks for this wonderful session. It was fantastic to see how the field is being energized, you know, building on some of the giants like Lynn Margulis and Carlos and people that have been working on this
goes.
Sounds like it's on now.
Yeah.
All right, just wanted to mention that it's fantastic to see this energy. And I think cohesive is a word that's also not forbidden term for seeing your efforts to push this field forward. I have become fascinated by Rotem Zorek's work, where he's looked at genes and gene systems that are involved in protection against phages and has discovered that many of these are actually present in the innate immune system of eukaryotes. And Yulia, I think it was you that brought that up, or many of you, you repeated the theme of that, we're not reinventing the wheel when we move from prokaryotes to eukaryotes. We're taking genes that were there and we're playing with them. And I was wondering how much you think or considered going back, looking at ancestral state reconstruction of proteins, of trying to follow these pathways more consistently so that beyond, for example, by coincidence, the work that Rotem did through looking at different phage systems, we could have a more cohesive picture of how our proteins have evolved.
Yeah, Tom, that sounds you.
Is this one on? Yeah, you can hear me. Yeah, it's a great question. So I think it's very possible to do that. I think one of the things that's very challenging in these ancestral reconstructions is balancing taxon sampling with the ability to actually do phylogenetic inferences. So microbiologists keep sequencing things constantly, and that's a huge problem for actually doing these analyses. But I think what's very possible now is to pick zoom in on the bacterial tree, and for a particular group, like let's say the proteobacteria, Pseudomonadota, it would be possible to reconstruct in great detail ancestral states and work out when systems evolved and how they recombined. I don't think there's been too much work on that yet, but it's certainly, we have the tools to do it. And yeah, there's also a lot of new work on trying to understand rules about how genes or how systems co-occur on genomes. And I think those could also be used in the ancestral context to do that much more accurately. So yeah, it's a cool idea for the future.
And then I guess I would add, once you've done it, it'd be possible to synthesize combinations to test some of these, right? So.
I wanted to thank you for a great session as well. I'm a eukaryotic cell biologist, and there's a lot of talk about biomolecular condensates, and I'm just wondering how you all think of biomolecular condensates in the context of evolution, particularly the pre-LUCA evolution and, consolidating enzymes for self-replicating entities.
Puri?
Well, this is an interesting area. Pre-LUCA, yes, there are people trying to do experimental evolution in vitro, and mostly related to RNA and imagining how some replicons can evolve and perhaps replicate, which is so far unclear because we don't yet know self-replicating RNA molecule. We only know molecules that cross-replicate, so that's a problem because if you have a conformation that is three-dimensional, you cannot self-replicate very easily. So people are trying. There is a huge field to explore. So that's certainly an avenue of progress.
Yeah, we have time for one or two more questions, so.
Hi, I'm curious what your views are. I don't think was mentioned on synthetic biology's kind of minimization model and kind of what its limitations are. It seems like such a hot field that gets a lot of press. And also, is anyone trying to minimize multicellularity right now?
That's a really interesting question. So I do know of efforts like the minimal cell that I think Jalen and others pioneered. And so you can really, I think if we can synthesize chromosomes, that would be really exciting to try to, and I think we're there. And so I think that there are these efforts. And I really do think that, there's a lot of work in synthetic biology on the regulatory networks and gene networks and trying to, I know Mike Elowitz is thinking about trying to figure out how you can basically develop regulatory circuits to get cell states. And so I think that's not a direct answer to your question, but there is, I think, we talked a lot in our colloquium about replaying the tape, can we use synthetic biology to resynthesize whole organisms and see if you can re-evolve and what happens, or what happens if you put a new gene function into an existing context. So I think there's a lot of potential.
I think maybe I'll just add one thing to that, which is that one of the things that Julie mentioned earlier was the importance of ecological context. So what constitutes the minimal life form is hugely dependent. I mean, it's completely co-varying with the sort of environment where we imagine that life originated. So that's an interesting facet to that whole discussion as well.
I had one more. So there's this book out there called From Bacteria to Bach. It's by the philosopher Daniel Dennett. And in it, he discusses this theory of molecular machine evolution called the Rube-Goldberg hypothesis, where it wasn't incremental. It was this kind of spontaneous complexity that emerged that was then streamlined. What are your thoughts on that hypothesis?
It sounds like you, Mike.
Yeah, I think the general target for selection is to make things as simple as possible. Just energetically, that makes sense, right? There's ways to break the organism as well, but organisms get into these situations where genes duplicate all the time, segments of DNA duplicate all the time, and in a bacterium, you can feel 2 or 3 more nucleotides in your genome, and they can be selected out. But in a eukaryote, it's diminishing returns eventually. You could insert a 10 kb piece of DNA, and energetically it would be such a small part of your energy budget, you'd never feel it, can't be eliminated. So I think a lot of complexity arises by effectively neutral mechanisms, and then you get this ratchet-like thing going on, and that's one of the arguments for why the ribosome became so complex. You get these sort of adventitious proteins that start to glue on to the side of your previous ribosome, and that covers up a part of the surface of the molecule, right? And that allows a more permissive environment for conditionally deleterious mutations to arise. That would be bad if you were exposed to water, but now you're no longer exposed. So I think a lot of the reasons complexity originates is for completely non-adaptive reasons. And you can do things with it later on, like introns did not originate for tissue-specific expression, right? They originated in the ancestor to LICA, and yet they are exploited for other reasons in our multicellular lineages.
Thank you.
I want to thank everybody, and we'll stick around up here if you have further questions. Thanks, everyone, for showing up around your lunchtime.